The topic of today's lecture, oxidative phosphorylation, corresponds to chapter 19 in Langer textbook. So what is oxidative phosphorylation? To understand its place in cellular metabolism, we shall go back to our discussion about cellular respiration. When talking about citric acid cycle, we introduce cellular respiration as set of catabolic reactions that allow cells to oxidize various complex organic molecules with atmospheric oxygen and use the vast release of chemical energy for cellular needs to produce ATP. When talking about cellular respiration, we staged it into 3 milestones or 3 steps and compared these steps to the function of the car engine, where the first 2 steps are used to prepare the car fuel, and the third final steps is used to combust this fuel with using atmospheric oxygen to release chemical energy that powers a cell. So from the chemical perspective, the first step, production of acetyl-CoA, is a way for cells to take a variety of chemically different complex organic compounds and convert them into uniform pieces, steel groups bound to a carrier molecule coenzyme A. During this process, this process, besides production of acetyl-CoA, releases some ATP and some reduced form of standardized electron transport carriers such as NADPH or NADH, and FADH2 and carbon dioxide. In the second stage, the uniform 2-carbon acetyl groups are oxidized in a citric acid cycle completely to carbon dioxide and produce some amount of useful cellular energy equivalents in the form of GTP and vast amount of reduced electron transport carriers in the form of, again, NADH and FADH2. Although the first and second step produce some amount of the useful cellular chemical energy in the ATP and GTP form, this is a byproduct rather than main product. And the main product are this standardized reduced electron transport carrier. In the third stage, these reduced electron transport carrier are oxidized by atmospheric oxygen, and this energy is produced to generate ATP, and hence this final stage of cellular respiration is referred to as oxidative phosphorylation. The oxidized forms of the electron transport carrier are then fed back into the first and second stage, thus allowing to this cycle of reaction in cellular respiration to proceed. The oxidative phosphorylation releases vast amount of ATP and takes place inside the mitochondrial matrix. The reduced electron transport carriers either produced from the TCA cycle that happens in the mitochondrial matrix, or channeled into mitochondrial matrix from other cellular compartments, are used as donors of highly energetic reduced electrons, and these electrons are pumped through the chain of complexes sitting in the inner mitochondrial membrane. They're called mitochondrial electron transport chain, and these electrons finally end up on highly affine electron acceptor, atmospheric oxygen, reducing oxygen to water. So the energy release from this produced by oxidizing the transferring, sorry, these highly energetic electrons to oxygen is used to power the proton pumps that the complexes of the mitochondrial electron transport chain represent, and the protons are pumped from mitochondrial matrix to outside of the mitochondria. This generates the electrochemical gradient consisting of um, the gradient of proton concentration per se and the electric field generated by disproportionation of charges between the mitochondrial matrix and the outside of the mitochondrial matrix. And this electrochemical gradients is utilized by proton pump ATP synthase, which is use the energy produced by releasing the protons back into mitochondrial matrix to synthesize ATP from the ADP in an organic phosphate. So the central to oxidative phosphorylation is this production of electrochemical gradient and coupling of electrochemical gradient to ATP synthesis, and therefore the theory that describes the inner workings of oxidative phosphorylation is referred to as chemasmodic theory. The learning goals of today's lecture is to understand how the mitochondrial electron transport chain functions to uh, couple the oxidation of the reduced electron transport carrier to the generation of electrochemical gradient, and to understand how the proton motive force built by electron transport chain is utilized by ATP synthase to produce ATP in the mitochondria. The topic of this lecture, oxidative phosphorylation, concludes our discussion about cellular respiration as such is based on the previous topics. Especially I would like to highlight the topics covered by Fergal, enzyme reactions and bioenergetics, and my previous topics, glycolysis and citric acid cycle.